So I would like to welcome you all to this Nansen Neuroscience Lecture for 2022. So my name is Lise Øvrås. I'm the president of the Norwegian Academy of Science and Letters, and it's a great honor for me to be here today and to welcome you to this event. Today we are celebrating Nansen's birthday. He was born October 10th in 1861, 161 years ago. When Fridtjof Nansen died in 1930, he had accomplished several achievements in highly diverse disciplines. He assisted refugees, went on polar expeditions, and got involved in diplomacy. It might not be as well known that Nansen also did important pioneering work within neurobiological sciences during the 5 years he lived in Bergen. Fridtjof Nansen started his academic career at Bergen Museum. He decided to study zoology, as this could be very well connected with fieldwork and outdoor activity that he loved so much. He was appointed a museum curator in zoology and soon became part of a highly active research group led by the leprosy doctor and zoologist Daniel Danielsson and the microbiologist Gerhard Armauer Hansen, who discovered the leprosy bacillum. Nansen was early interested in organization of the nervous system and primitive marine animals collected at the Norwegian North Sea expeditions. During his years in Bergen, he learned advanced methods for neurohistology. At the age of 24, he wrote the first scientific paper printed in the annual report of Bergen Museum. For his debut work, he received the gold medal at Bergen Museum, the Joachim Friele Gold Medal. He decided to exchange this gold medal to acquire travel money to go to Germany and Italy to learn Golgi's method for silver staining of nervous cells. He provided impressive drawings of the nervous system, demonstrating that these were made up of a separated network of ganglion cells connected by synapses. 2 years later, in 1887, Nansen handed in his PhD thesis, The Structure and Combination of Histological Elements of the Central Nervous System. A solid piece of work where he acquired the knowledge about these histological techniques of cross-sections of the animal, followed by beautiful drawing illustration made by Nansen himself of lithography on stone plate, which later was printed in Germany and which today is available to download digitally for everyone interested, and I would recommend it. They are beautiful. Retrospective, Nansen is recognized as one of the forefathers of the neurotheories based on his PhD work. This represents today one of the most important classic contributions to biological sciences. On September 21st, I was attending the Carnegie Lecture at the National Academy of Science in Washington, D.C. This meeting was a conversation with the Kavli Prize laureate from 2012, Corey Bergman, and the science reporter Frank Sesno on the topic, One Brain, Many Behaviors, the Fascinating World of Circuit Neuroscience. During this conversation, they discussed how genes and the environment interact to generate a variety of behaviors, and how behavior decisions are modified by context and experience. In 2022, neuroscience has reached a stage where these questions can be asked with great precision, and in some cases answered. But for some of these very complex questions, we might still seek answers from primitive animals, such as Nansen did in 1885. The Bergman Lab has studied these questions in the nematode C. elegans, whose simple nervous system consists of only 302 neurons, yet the animal can move around, explore the environment, evaluate the social context, and learn from experience. What I found very interesting and fascinating is that the animal and human behavior is not fixed, but is reversibly modified by internal motivational and emotional states. Today's topic for neuroscience lecture is aging, which is a very timely and fascinating topic, and I look very much forward to hearing the answer to your proposed question, is aging inevitable? Congratulations with assembling a very interesting program, and I hope you all will enjoy today's Nansen Neuroscience Lectures. Thank you.
So welcome everybody, it's nice to see you. And everybody that is interested in physical exercise They will have a great time today. And I'm very proud that today's lectures will be given by Henriette van Praag. She's one of my leading stars, at least, when it comes to science. So Henriette van Praag, she's actually from the Netherlands. You're born there and you grow up there. But later on, you became an associate professor of biomedical sciences at the Charles Smith College of Medicine in Florida Atlantic University. And you've been there since 2018. And you serve as a co-editor-in-chief for the Brain Plasticity Journal, which is very, very nice journal. After completing your graduate studies in Tel Aviv University in Israel, you carried out postdoctoral work at Robert Wood Johnson Medical School in New Jersey, and you also been a staff scientist at the Salk Institute for Biological Studies in California. And before you joined the Florida Atlantic University, you have been a principal investigator at the National Institute on Aging, important here, in Maryland. Your laboratory and your investigation study how exercise affects structural and functional plasticity of brain areas that are important for learning and memory. And a lot of your people being here in the audience know that this is about hippocampus. So please, Henriette, the title of your talk, The Benefits of Exercise for Brain Function.
Thank you very much, Linda, for that kind introduction, and I'm very honored to be here today and speak on Nansen's birthday. So as you all know, neurons are essential for cognitive, emotional, motor, and sensory function. And indeed, as stated by Nansen himself, one of the original proponents of the neuron doctrine in his thesis in 1886, the more the intelligence of an animal is developed, the more intricate becomes the web of plating of nerve tubes and fibrillae in its dotted substance. So this is true, but unfortunately, with aging, there is also sometimes the loss of synapses, and with neurodegenerative conditions, even the loss of neurons themselves. And this is particularly true for the hippocampus, a brain area that is essential for memory function. It's one of the first brain areas that is affected both by normal aging and by Alzheimer's disease. However, the good news is that this brain area also has a high level of plasticity. For instance, it can give rise to new neurons in the dentate gyrus subfield in mammals postnatally. However, for about 100 years ago, the reigning dogma was that no new neurons could ever be generated in the adult brain. Once development was ended, the axons and dendrites dried up irrevocably, was stated by Ramonica Hall at the time. However, we now know that this is not quite so. There actually are two neurogenic zones in the adult brain. The subventricular zone of the lateral ventricle can give rise to new neurons in the olfactory bulb, and the subgranular zone of the dentate gyrus of the hippocampus can give rise to new granule cell neurons. But this whole field was a bit of an uphill battle. Altman and Duss indeed published using tritiated thymidine that there were in the postnatal brain, there were labeled cells, but they could not conclusively prove that these were actually neurons. They did not have markers to co-label with tritiated thymidine. They just said there's absence of glia in the layer, so that's why these are neurons. But the work was not, even though it was published in high impact or high profile journals, it was not accepted. And then 10 years later, Michael Kaplan did similar work using tritiated thymidine, this time combining it with electron microscopy, showing that these cells actually have synapses. But again, his work was also ignored, and he went back to practicing medicine. So what then revived the interest in this field? This was seminal work by Fernando Notenboom in the 1980s and early 1990s in the avian, in canaries, and also in food storing birds. And he showed when he captured birds that were free ranging and injected them again with tritiated thymidine, that they had higher levels of thymidine incorporation towards the fall, so when they were getting ready to store food, suggesting that these cells, first are generated in the adult and might even have a role in memory function. So this then generated the interest again, or sparked the interest by mammalian researchers to take a look at this again. Plus, around this time, there was new methodology, or better methodology. There was bromodioxyuridine labeling, and there was an antibody now for to label BRDU, so that you can see here. This is a coronal section through the mouse dentate gyrus. The BRDU positive cells are red. Now there's a neuronal marker in green, and if you see cells where there's an overlap between these two markers, now one can infer that these are indeed new neurons that are being born in the adult brain. But still, this was not enough to convince the general scientific community that indeed the adult brain could give rise to new neurons. Because they said, well, BRDU could maybe also be taken up by cells undergoing DNA repair. So what to do about that? You'd have to prove that these neurons are actually functional, that they have not just a nucleus, but they also have axons, dendrites, that you can make electrophysiological recordings from them. So we set out to do this, making a high-titer retrovirus expressing GFP, which we injected into the dentate gyrus directly of the hippocampus. And this showed really the progression, morphology, and physiology of these neurons, that they are really real neurons. And so, oops, sorry, little jump here. So then a colleague showed that you can also record from these cells over time following their physiological development, and also that these cells then express different markers, starting with nestin, SOX2 at early on, followed by PSA, NCAM, double court, and finally NUN and calbindin during the course of their maturations. So functional new neurons do exist in the adult mouse hippocampus, and the adult rodent hippocampus, and in many other mammalian species, potentially even humans. And the other unique fact about adult hippocampal neurogenesis is that it's highly plastic. It can be upregulated and downregulated. Upregulated by exercise, downregulated by aging, stress, high-fat diet, excessive alcohol intake. And this all happens against a background of different genetics, growth factors, hormones, neurotransmitters. So the original discovery of how did that come about, that we found that exercise increases neurogenesis. So there was a paper preceding me by Gerd Kemperman and colleagues showing that if you house animals in an enriched environment with more toys, more friends to play with, there's more neurogenesis. I sorted out these different components and found that indeed exercise is the critical component for the production of these newly born neurons. And you can see my design was not that good. I had different shapes of cages, but then when I had my own lab, I was fortunate to get a postdoc who did a much nicer job than me and put everybody in the same size cage, standard wheels, toys only, and the enriched environment with the wheels. So basically sorting out these variables in even more detail. And what we then found very convincingly is that Only when running is included is there an increase in cell proliferation and cell survival in the dentate gyrus of the hippocampus. And you can see it again here now with combining BRD with nu N, that the critical component is running. And this is also accompanied by an increase in BDNF protein in the hippocampus, which will support probably the survival of and integration of these newly born neurons. So exercise increases adult hippocampal neurogenesis. We also now know that this increase with running happens mainly in the dorsal dentate gyrus. So here you can see now dorsal aspect as the largest increase in these adult-born neurons. And the location of neurogenesis is important because we see a close association between improvements in learning and memory and adult neurogenesis. And the dorsal hippocampus has been traditionally associated with cognition, whereas the ventral aspect is more related to mood and emotion. So running in a running wheel improves learning and memory. And you can show this very clearly in the Morse water maze, task that you are probably all familiar with, where an animal learns to find a platform hidden under the surface of a pool that's made murky with a little bit of paint. And regardless if you start the animals running at a young age, middle-aged, or aged, after 1 month of physical activity, they will improve their performance on this task. So with that, we showed that exercise enhances performance on general hippocampus-dependent tasks. But as you all know, the hippocampus consists of multiple subfields. And if you make a lesion in any of one of those, it will affect Morse water maze learning, for example. So we looked for a task that was more specific to the dentate gyrus. The dentate gyrus is considered to be important for pattern separation, our ability to differentiate between highly similar events and stimuli. A neuroanatomical basis for that is that the entorhinal cortex has fewer neurons than the dentate itself, allowing for a dispersion of information and thereby finer encoding the separation of information. So then we asked, does running improve the ability to distinguish between highly similar events or stimuli? And for this, we use the so-called touchscreen system, where a mouse learns to touch the screen of a computer, an icon on the screen for a reward that comes out here, a sweet drink in the back here from a magazine. So we asked, is enhanced neurogenesis associated with improved pattern separation? And so the animals were trained to touch the screen, then subsequently housed with the wheels, and then tested in a probe trial or sort of final exam whether they could distinguish which one of these two icons, either in the big or small conditions, were associated with reward. And what we found at the time was that runners outperformed controls in the acquisition of the small but not the big separation. So when the task was more difficult, more challenging, the runners needed less trials to criterion, and there was an association between the trials to criterion and the number of newly born neurons produced, suggesting that running enhances spatial pattern separation. And this behavior is one that is also very much studied in humans' aging studies, because pattern separation is one of the first signs. If that goes, that is one of the first signs of a memory impairment and cognitive decline. So it's a rather labor-intensive task to train the animals on this, but it's important because it's very translational. So exercise benefits memory function, and adult neurogenesis may be one of the underlying mechanisms. But would it be enough to just, let's say, if you had a magic pill and increase the number of new neurons in the dentate gyrus, would that be enough to enhance learning and memory? Or are these neurons really interconnected throughout the brain, and do you need to not just add new neurons, but also change the entire network? So that's another question that we asked in the lab. So we knew from our electrophysiological recordings that there are synaptic inputs to these new neurons for both GABAergic and glutamatergic. But where were they coming from? And are they also modified by exercise? So to identify the direct inputs to new neurons and their modification by running, we used a combination of a retroviral vector, now expressing the TVA receptor, a receptor that's foreign to the mammalian brain, and we combined that with rabies virus tracing, a method also that many of you are probably familiar with here. But what is nice about this method that you can label these newly born neurons, and then you can wait a few days, a few months, and actually we now have a study actually even going out for a year after labeling the newly born neurons with retrovirus. And then you can, at those time points, get a snapshot of what the circuitry looks like of these newly born neurons throughout the lifespan of the cells and throughout the lifespan of the animals. And it's very dynamic. It changes over time. So you can see this is the basic development of these newly born neurons. As I showed you earlier, this is like when they're a week old, 14 days old, 30 days old. So, so this method allows you to ask many questions. For example, there's a reigning, again, a sort of a dogma in the neurogenesis field that the adult-born neurons, similar to neonates, first receive GABAergic and then glutamatergic synaptic inputs. So this was something, for example, that we could address with this method. And we showed that actually, no, both glutamatergic and GABAergic inputs to adult-born neurons come in when the cells are only 1 week of age. So you can see pyramidal cell input, mossy cell input, interneuron input, different types of interneurons that provide input to these newly born neurons, neuropeptide Y, somatostatin, parvalbumin cells, and also some input from mossy cells, which are glutamatergic, and from pyramidal cells. And we then further supported this work by applying NMDA to acute slices of retrovirally labeled, new neurons in them. We see that there is a tonic current that comes up with NMDA application. And also when we stimulate the granule cell layer, we can evoke both glutamatergic and GABAergic currents in these very young adult-born neurons. And we've also further supported this by optogenetics, injecting AAV-expressing channel rhodopsin into the dentate. And now the newly born neurons are here in red, so expressing RFP. And you can see again that they are responding with synaptic responses to the stimulation, and that this can be blocked specifically by glutamatergic antagonists. So basically this method allows us to study how the inputs to new neurons evolve during their maturation, when do they come in. As you can see, the entorhinal input only comes in when the cells are about a month old. And how does this evolve both over the lifespan of such neurons and over the lifespan of the animals? And how does it change with exercise? So one of the things we asked, does running change the input to these newly born neurons? So the same approach, control, running, retrovirus, rabies virus, and then we look a month later. So this is, I don't know if you can see, maybe you can reduce the lights a little bit in the front, but you can see the input from the lateral entorhinal and medial entorhinal cortex thank you very much, onto these newly born granule cells under control conditions. So these red cells are not newly born neurons, these are trace neurons. So the granule cells are in here. But with exercise, there's a huge recruitment of the entorhinal cortex through the peripheral pathway. So you can see really this thick band here of the lateral and medial peripheral pathway that you see only really under running conditions and not under sedentary conditions. And this is complemented by behavioral work where, for example, if we take out the lateral entorhinal cortex, we see an impairment in pattern separation ability. So you can see that here the animals need more trials to criterion. And again, conversely, with running, as I showed you earlier, we see improved performance in the touchscreen task. And we've also, not just showing you the pictures, but we've done also electrophysiological and also detailed histological work, quantifying these inputs, and for example, showing that under sedentary conditions, the larger input is from lateral entorhinal cortex onto these newly born neurons. So, so what I've shown you so far is that running increases neurogenesis, but it's not just that we have more of these newly born neurons. It's also the case that running actually rewires the circuitry of these newly born neurons, so their inputs change with exercise. So it's not just that the dentate gyrus of the hippocampus is not static. The entire brain has a large degree of plasticity in terms of the connections that are formed. So running increases adult neurogenesis, network plasticity, and learning and memory. But we still don't know why. Why does exercise improve cognitive function? And I've just shown you only one thing that happens with exercise, and that's, or maybe two, increased neurogenesis, circuit modification, and upregulation of BDNF. But as we well know, there's also extensive effects on neurotransmitters. There are effects on fine morphology, such as changes in spine density. There are effects on LTP, synaptic plasticity, multiple growth factors, and there's also changes in brain blood flow. Actually, those can also be measured in humans with exercise. And we also see changes in the vasculature. We see changes in angiogenesis with exercise. So there are many things that happen. Why do these happen? Well, one of the ways we try to address this is to look outside of the brain. For example, is there a link between muscle and brain? Because obviously with exercise, the brain is not static. There is a communication between the periphery and the brain. So, so what is the basis for this concept? There are several what we would call maybe converging pieces of evidence. One based in heterochronic parabiosis. This is a very nice paper by Salvaleda and colleagues where he connected the bloodstream of aged animals to young animals and showed that this resulted in a decrease in adult neurogenesis. Subsequent work from himself and also from his former advisor and colleagues shows that if you take plasma from running mice and you infuse it into sedentary mice, this improves memory and enhances neurogenesis. And I know this type of work here in Norway is being pursued even in found the effect on learning and memory is abolished. So if you activate muscle AMP kinase with ACAR, you can enhance neurogenesis and memory. And this suggested to us that the effects of exercise for cognition could originate from muscle. And then we obviously wanted to know, well, if this is the case, well, which factors are then released into circulation by muscle AMPK activation that might affect brain plasticity? And for these studies, we went in vitro using muscle cell cultures, and then treated these L6 muscle cells with either vehicle or ACAR, and then collected the conditioned media. So this was work done by Hayo Moon in my lab, who's now a professor at Seoul National University in Korea, South Korea. So collected the media, and then he took neural progenitor cell cultures, which are multipotent in vitro. So this is the proliferative undifferentiated state, but you can add different factors and you will have cells that become a neuronal phenotype, oligodendrocytes, or astrocytes. And he took the muscle conditioned media and put it on such cultures. And what he found was that if you take muscle conditioned media, some conditioned media meaning the cells were treated with ACAR, you put it on the neural stem cells, then you'll have an upregulation of this neuronal marker. Now, for all transparency, it's not just actually neurons that become more pronounced, it's also glia. So you can have basically an increase in neuronal and glial differentiation if you place this media on these neural stem cell cultures. But what are the factors that are released in this media that are responsible for such effects? So for this, he collected the conditioned media, concentrated it, and compared it on a gel here, and looked at the differentially expressed bands, excised them, and we sent them for mass spectrometry. We got several candidates back, and based on criteria such as the size of the protein, whether it could be secreted or not, and previous arrays, we selected Cathepsin B as our most promising candidate to be a myokine. And then went back in vivo and found that Cathepsin B is upregulated in muscle, so gene expression increases with exercise, 1 month of exercise, so upregulation of protein, and there's an upregulation of Cathepsin B levels in plasma following 2 weeks or 1 month of physical activity. Then when we studied is there behavioral relevance for Cathepsin B, so we were fortunate to obtain Cathepsin B knockout mice, housed them either under control or running conditions, tested them in a battery of behavioral, motor, mood, and memory tests, and we found that running no longer enhances spatial memory in the knockout mice. So you can see the wild-type mice and do well in the probe trial, but not so for the knockouts. In addition, there is no longer a neurogenic effect of exercise in these animals. And this is supported by some of our work in vitro again. Here we apply the Cathepsin B recombinant antibody to the hippocampal progenitor cell cultures, and we see that it increases the expression of double cortin, an early neuronal marker, and also BDNF. Then Moon asked, well, does exercise have any effect on in humans? Does it increase with exercise? So here we were fortunate to collaborate with Emra Duzel in Thane Magenburg. We had control couch potatoes, walking twice a week. These were students, young adults, and then treadmill training, 70 to 90% heart rate, max heart rate for about an hour for 4 months. And in this case, we found that increased fitness is associated with elevated plasma levels, Cathepsin B plasma levels in humans, and also that if you challenge people with quite a difficult memory task, I think, is the Ray Ostrich complex figure task, where you have to, you get this picture and you copy it on a piece of paper. The experimenter takes the paper away from you, and then you have to draw it again from memory a half hour or an hour later. And it's not that easy, but those people who were exercising and had a good increase in their Cathepsin B levels did better on this task. So another human study that we were fortunate to collaborate with pertained to aging and Alzheimer's disease. There's some controversy as to whether Cathepsin B is either beneficial or detrimental to amyloid production and accumulation. Some papers say that it has anti-amyloidic and neuroprotective effects. Others say it increases it. But we are on the side of, in this case, of the beneficial effects of elevating Cathepsin B, and I'll show you why. We collaborated with Professor Oziomo Okonkwo at the University of Madison, Wisconsin. And we asked, does exercise increase Cathepsin B levels in older adults who are at risk for Alzheimer's disease? Is it because they have a parental first-degree relative with Alzheimer's disease, or because they have ApoE4 positivity? Their average age is 65 years old, and their exercise intervention was, again, long, for 6 months. This is a clinical study called the REACH study, Aerobic Exercise and Cognitive Health at the University of Madison. So people had either usual physical activity, like walking your dog, or they were training on the treadmill with increasing intensity 3 days a week, up to 70 to 80% of their max heart rate. They showed in a previous study that this is associated with improved cardiorespiratory fitness, and also there are some cognitive improvements in this small cohort. And again, so we collaborated with them, and they sent us the blood samples, and we looked at the Cathepsin B levels and BDNF levels in the plasma of these people. Somebody else also did the aging factor Clotho, and then there was also a metabolomics analysis performed. And what we found was that exercise increases plasma Cathepsin B levels in older adults that are at increased risk for Alzheimer's disease. You can see here so the triangles are the males, and the circles here are the females. Females being particularly responsive in this case. And we also ran a correlation with the cognitive testing that was done before and after the exercise intervention. And this is not the Ray Ostrich test this time, but it's the California Verbal Learning Test. So basically, how many words, a list of words that you can remember. And again, there's a correlation between changes, the delta of Cathepsin B, and performance on this task. And this was not the case. This correlation was, we only saw it with Cathepsin B, not with BDNF or with the other marker we looked at, Clotho. And then finally, in this study, we were fortunate to also be able to do some what we call metabolomics. So these samples were sent to a company called Metabolon. And what came back from there is that plasma lipid metabolites, such as sphingonines and sphingosines and ceramides, that are associated with the transition from what we call minimal cognitive impairment to Alzheimer's disease are lowered by physical activity. So this was consistent with the other findings in the study, the improvement in cognition, the increase in Cathepsin B. And very interestingly, here we see a strong correlation with the changes in BDNF. And again, this is another such cohort of lipids that are reduced by physical activities, in this case phosphorethanolamine, for example, and that again have been implicated by others in this conversion stage from MCI to AD, and they are lowered by exercise, potentially preventing or maybe a biomarker that may indicate how we can prevent or delay the onset of disease in these cases. So in this study, so enhanced physical activity in middle-aged adults at risk for Alzheimer's disease, we see that there are increased plasma Cathepsin B levels. There's a correlation between Cathepsin B and memory. Plasma BDNF was lower, unchanged clotho. I told you about the lipid metabolites and the correlation with BDNF. But we think that this may be a way, this sort of more comprehensive analysis to get some better handle on blood biomarkers that could be either predictive for disease or that also could tell us which exercise interventions, how long, the intensity, duration, age of onset to get the most out of exercise interventions. So altogether, looking at the underlying mechanisms of how to increase neurogenesis, synaptic plasticity, we have to take ourselves as neuroscientists also outside of the brain, look at other organs such as muscle, adipose tissue, liver, I think to better understand how physical activity enhances synaptic plasticity, neurotrophin levels in the hippocampus, and improves neurogenesis, cognition, and mood. So in conclusion, running increases, I hope I've convinced you of that, running increases neurogenesis and memory function. It also vastly changes not just the number of cells, but also the network of these neurons. Some of the benefits of exercise for cognition may originate from the peripheral organs. One such factor I talked about is Cathepsin B, but every day there may be new factors that are discovered that can influence the brain. And with that, I thank you for your attention. I'll be happy to take questions.
Thank you very much indeed. Yeah. I think I can fix it, but we have to turn the pole. Okay. Okay. So that was many different things, and it is not a wonder that muscle and brain are interconnected by many different factors, really. I think if you think of what happens and has happened always for individuals to survive in the hostile environment, you must need muscles and brains. And we can, as you mentioned, we can expect that there must be much more to discover, and also how it really interrelates. I would like to see some questions, and Professor, yeah, David.
It's a really wonderful talk.
Thank you.
I'm not sure if this is an answerable question, but I was wondering, whether what you, in a way, are describing is a deficiency produced by modern life in terms of cognitive function. So, I asked myself, why is exercise producing this beneficial effect? Is it because, in a way, we've evolved to live in an environment where normally we'd be highly active, so that, in a sense, in our kind of ancient natural state, there wouldn't be any benefits of exercise because we'd all be walking around everywhere. Or alternatively, whether what you're looking at is, at least to some extent, not just a deficiency in exercise, but an enhancement effect. And I guess the question there would be, what's the dose relationship between exercise and cognitive function? Does it sort of fit the idea that you're just restoring something that's missing because of modern lifestyle? Or actually, do you get a continual sort of increased performance so that you're really getting an enhancement effect, kind of above the normal from excess?
Yeah, that's a good question. So, of course, how far can you push the system? That is a good question. We don't really.
Is it like vitamin deficiency? I suppose you could put it that way.
I'm sorry?
Is it like vitamin deficiency?
Yeah, in a way. So there's a lot of studies showing that really sedentarism or lack of physical activity, leads to cognitive hypofunction. In animals, if you give them a high-fat diet and have them be sedentary, there'll be lower levels of BDNF, lower levels of neurogenesis. So basically, yeah, you're right, it is a deficient state. Also, as I mentioned in our circuitry analysis with our rabies tracing studies with exercise, we see that in the sedentary condition, so this is some unpublished data where we look over the lifespan of the animals, there's like some of the structures sort of drop out. The wiring sort of becomes more spotty, whereas if you live the continuously active lifestyle, so not just, intermittently, but active throughout the lifespan, the wiring really stays. It's not identical to what we see in the younger animals, but it remains more complete. So yeah, it's a very good question. But how far we can optimize, I don't know. Yeah, right. Yeah.
Evandro Feifang.
Thank you, Professor van Praag, Evandro Feifang from University of Oslo. And I actually have known Professor van Praag for 10 years when I started my postdoc in NIA. And my office is 2 meters from Professor van Praag. So I have followed her research for many years, and I was very impressed in the past 5 years. You got so many results to support the hypothesis. I'm especially interested in the Cathepsin B story, because it is a protein at least involved in lysosomal function. And it has been shown it is the last step of autophagy, a way to cleave the brain garbage. So studies from us and others have been shown impaired autophagy contribute to Alzheimer's disease.
Right.
So I wonder, in your case, whether exercise can help to increase autophagy while increase lysosomal activity as one of the approaches to reduce cognitive impairment in your models. Thank you.
Yes, thank you, Evandro. And yes, we had a very nice time at NIA, and it was a pleasure to have you there as a colleague. Yes, I think you're right. Increased autophagy may be one mechanism by which you could, rejuvenate cells and the brain by clearing out, deficient proteins or accumulation of lipids in the cells and increasing maybe mitochondrial biogenesis. So I think that's a very good approach. In the case of Cathepsin B, obviously it has a lysosomal function. And, but it also has a secreted function. So it is a protein that has multiple ways of exerting its effects. So one may be, as you say, similar to autophagy, but it can also, this work by others, not by us, showing that it can be, extruded through the lysosomes to the extracellular space, and there it will affect the extracellular matrix, the structure of the perineuronal nets, and thereby rejuvenate synaptic plasticity, because it will allow for the increase in spinogenesis, in dendritic spine growth. So it is, I would say, a pleiotropic molecule in that sense.
Question here.
Yeah, sure. Thank you for a very brilliant talk. My name is Alexander. I'm from University of Oslo. So my question is, in one of the studies you use icar, the
Yes.
drug, and what do you think was the translational perspective of this drug? Since I'm wondering, because I know that this drug actually was prohibited by anti-doping agency, and it's quite interesting actually, like in what kind of fields we could apply this drug. Yeah. So what do you think about it?
Yes, yeah, it is on the. You're right. It is forbidden for athletes to take ACAR, though I think it is done sometimes. And actually, we would warn very strongly against the use of this compound, because what we see with, especially in young animals, with prolonged use, it is actually becomes detrimental for learning and memory, and it will you will see an upregulation of IL-1, of inflammation, and also we've done microarray analyses, so after 7 days of use in young animals, you'll see an upregulation of genes important for synaptic plasticity in the hippocampus. After 14 days of use, you will see a complete reversal. So the compound is only beneficial for a short window of time, and not for prolonged use. And this you could, if you're thinking in translational terms, you could think it maybe as, maybe overstimulation, or what you would happen, if you run like, 5 consecutive marathons, which would also upregulate inflammation and potentially be detrimental for the brain. So I think the therapeutic window is narrow for this compound, but it's not absent, and especially with aging, when there's less when there's less alpha-2 receptors already in the muscle, you can give it a little bit longer, and it may benefit as sort of a crutch drug, between getting sedentary people back up out of bed or something like that. But good question, yeah.
Thank you for that. Okay, thank you for a nice presentation. Very important direction of development for us aged people, as I'm 86 years already. But I'm Thomas Böhmer from University of Oslo. We have an important remark yesterday about your marathon training, as we have a Norwegian won the world competition at Hawaii. He was running 7 hours that day, and he has been training really from when he was age 12 to now is 26. But that's not usual for us, most of us. But what about, you were speaking about high cardiovascular training, but what about muscle training, which is also advised for us aged people?
Yes, so that's again a good question. So resistance training is generally in the human studies used as a control, like light weights or stretching or things like that. But some people have looked in rats by putting little weights on their tails and having them climb up ladders, and then looked at changes in proteins and gene expression in the brain. And they see some effects, for example, on IGF levels in the brain. However, the bulk of evidence supports aerobic activity for cognitive health. And this doesn't have to be marathon running or high-intensity interval training. It can just be, a daily walk, as long as it is above your own base level. That said, there is a link between what we call sarcopenia, or muscle frailty, and cognitive decline. So this suggests to me also that maintaining muscle strength is very important in aging. And that will also help the walk. So I'm very much a proponent of lifting some weights.
You have a question?
Thank you. I'm Britta Helleborg, and I'm really happy to be here listening to your talk. And I was thinking, this exercise thing, when we get, is it only bringing new neurons while running, or is it last for minutes, hours, days thereafter?
So the new neurons, when they are generated in the rodent brain, they there is an initial phase of what we call cell proliferation. So let's say 100 cells are produced on a given day, but then not all of them make it to the, to the finish line. So let's say 50 survive, but if they survive, if they pass that 2-week time point of critical point, they will survive generally for the lifetime of the animal. That's why we can also do the retroviral labeling and look at circuitry so much later. So those cells that are generated, they are there for good. And that's also why we think that being active when people are young, or when the organism is young, when there is a great deal of plasticity and more evidence for postnatal neurogenesis also in humans in the teenage years, that is really important to get the optimal benefit, as my colleague was mentioning in his question, is in youth, because those changes, those cells that are generated then probably give you a lifetime, sort of reserve bank that lasts into old age.
Thank you for a very nice talk. I was wondering, the mechanisms that you show, do you think they're general for all the areas of the brain where neurogenesis occurs, or are they specific to the hippocampus?
So yeah, that's a very good question. Actually, one that we addressed sort of early on when I came up with the first exercise study. I had taken some sections of the brain, also through this lateral ventricles, and I gave them to a colleague, to Georg Kuhn, and he analyzed them, and he said, no, there's no effect of exercise in the subventricular zone. But others since then, I think out of Cindy Moreland's group has suggested there, and also a group in Australia suggests that there is some effect of exercise also in that stream. But I think what I think the more robust effect is probably for hippocampus. But there are other non-neurogenic areas that benefit from exercise, such as the frontal cortex, which is important for planning and executive function. That will show changes in growth factors or volume with physical activity. So the effects of exercise are not restricted to the hippocampus, but it also affect other brain areas that are not necessarily neurogenic.
Last question here.
It's very brief. First of all, thank you so much for the talk. My name is Maria, and I'm a postdoc in Evandro Fan Lab. Actually, my question is as well related to Cathepsin B. I guess it's because we are interested because of autophagy and lysosome pathways. And I wonder, because I know with Cathepsin D, there is a bit of controversy too. And my question is, if you know if that the reason of these controversies could be the way of destruction, because I know when you study in blood some of these markers, some of them are in exosomes, and maybe depending on the method, you can get different results. So,
you mean the method of analysis?
Yes.
Yeah, we have not studied, for example, whether Cathepsin B is packaged in exosomes or in other aspects. But yeah, that is very possible. That how you look at the protein, you will get different results. So, but Cathepsin D, I think, is more associated with detrimental effects in terms of.
Thank you very much, and thank you to the audience. And now,
thank you.
Okay, then we go on. Our next speaker, his name is David Jones. He is a professor of biogerontology at the University College of London. And he is working in the Institute of Healthy Aging, which fits the topic for today. He's also a founder member and a research director of this Healthy Aging Institute. He studied biochemistry as an undergraduate at Sussex University and then genetics as a doctoral student at Glasgow University. And as a postdoc, he worked on nematode parasitology at Imperial College London, before moving to the University of Missouri Columbia in the States to work on aging together with Professor Don Riddle. He returned to the United Kingdom in 1997 to start his own research group at the University College of London with the Royal Society University Research Fellowship. The aim of his research are to understand the causes of aging and identify general principles of pathophysiology or late life diseases. Much of Germ's work uses the nematode C. elegans, but he also contributes to studies of aging of other nematodes, fruit flies, and the mice. And he also penned articles on aging and ethics. And I have to tell you that he's been very instrumental in our course given in December about biology of aging and giving a lot of very nice advice how to put this team together. So please, David.
Thank you. Okay. Is that the clicker, this one? Yes. Well, thank you very much to the organizers of this meeting. It's an honor to be able to present something in this session on is aging inevitable? So I'll be talking more about the aging part of it than the neuroscience, really. So I'm going to start my talk with reference to death and the causes of death, why we die. I suppose that in the last year or two, one particular cause of death has been very much uppermost in our minds, the COVID epidemic, and which is sort of on the wane now, it seems. But now we have a new cause of death in the form of Vladimir Putin. But in terms of real, numbers of actual deaths in the world, we can see here a representation of what we all die from. So this is showing you the each of the major causes of death in terms of how many relative deaths it causes in a sort of a hierarchy. You can see this is cardiovascular disease, so things like stroke and heart disease and cardiac arrest and so on. Cancer is the second major cause of death. Respiratory disorders, so this would be things like chronic obstructive pulmonary disease, the third major killer. Nervous system disorders, which Alzheimer's disease would be included there. Digestive disorders, kidney infections, of course. Elderly people are much more prone to infection, so this mainly affects elderly people. And in fact, overall, as you go down, except maybe to the very bottom with pregnancy and war and so on, it's mainly aging, which is the risk for these diseases. And I think one can say with sureness that the main cause of death, the main cause of chronic and lethal illness in the world today is now aging, which is a biological process, the nature of which really remains very poorly understood. So even though I've been working all these years on aging, I know the field very well, and I know it well enough to be able to say that this is a process that we really, really don't understand yet. A simple reason for that is that it is a very, very complex process. And so with very complex questions in biology, one approach to try to solve them is to try to find a really simple organism that will exhibit the biological process that you want to understand, and try to understand it there where things are easy. And that's why I work on this organism, Caenorhabditis elegans, which is a nematode with a lifespan of only a couple of weeks. And it's a very, very easy organism to work with in the lab. So if we could understand aging in terms of its fundamental principles, what actually is aging? What actually causes it? Where do all these diseases come from? Why do older people get Alzheimer's? We really don't know the answers to those questions. What are the mechanisms? If we could understand that, then we could take the knowledge that we have and apply it to an organism that we actually care about ourselves and understand the origins of aging-related disease. So understanding aging in C. elegans is very, very important strategically. So what I'm going to do in the talk today, I'm going to start with sort of a very kind of high-up, broad brush introduction to the field as I see it at the moment. So it's almost like I'm going to give you the conclusions at the beginning in a very broad brush way, which includes what I view as really good news for the aging field, which is what I believe is the beginning of an emerging paradigm, like an actual, what may be the sort of earliest version of a mature understanding of aging. And this is really something that's emerged in the last few years. The second half, I'm going to talk actually more about actual worm biology and give some sort of bad news about the likelihood of some major interventions in aging, which seemed possible a couple of decades ago. They seemed like this might be a possibility, but I think the prospects for that, for human aging, are actually looking a lot worse now, and I'll explain why. So, having worked for so long on aging, this is a question that's started to trouble me increasingly from about 15 years ago, realizing that the field kind of wasn't getting the profound answers that it needed. So why don't we understand aging? In a fundamental way, the field of the biology of aging lacks something that's present in more mature fields of science, such as chemistry. So chemistry, in a way, reached maturity as a field with the development of an understanding of the nature of the chemical bond and the development of the periodic table and a number of other sort of aspects of fundamental understanding that produced a science of incredible power. Once you've got that fundamental understanding, you can move forward with a terrific speed and power. So in genetics, you have the discovery of Mendel's laws, and then in molecular biology, the central dogma of how genes function and so on. And neurobiology, I mean, the Professor van Praag's talk, that kind of work you could not do in aging because of the knowledge that exists about neuronal function. In aging, we simply don't have a paradigm of that sort. So I think in many ways I was struck by sort of thinking about the similarities between aging and the kind of age of polar exploration from Nansen and other explorers like Roald Amundsen. And the fact that in a way the aging field lacks, there's this great unknown continent of understanding. Perhaps similar to, the last talk made me think of the question of the mind-brain and how you could understand consciousness in terms of neuronal function. It seems like we're not anywhere near that. Yeah? So these are sort of continents of the unknown. And I think one of the reasons why things haven't moved is because of a sort of a working paradigm, if you like, that dominated the aging field for many, many decades, which is this, what I call the damage maintenance paradigm. I'll come back to this a bit more later. But the idea that damage accumulation is fundamental to aging. And I think that certainly I've worked on this for about a decade, testing this in C. elegans, and this model really seems to be quite inadequate as an understanding. Though it may play a minor role in some aspects of aging, it doesn't seem to be the main thing, especially not in C. elegans. It most certainly isn't. So if I can, I'd like to sort of draw a certain analogy between the damage maintenance paradigm and the choice by Robert Falcon Scott to use horses to try to reach the South Pole. And I mean, in a way, this was he wasn't going to succeed in coming back alive with this poor choice of way to go forward. So with a paradigm that doesn't allow you to get there, you won't get there.
So I think one of the things that's happened with the kind of collapse of confidence in the damage maintenance paradigm is the appearance of something called the hallmarks of aging, which is something that's very often name-checked at the beginning of talks, which is really something that in a way has the appearance of a paradigm and I think is mistaken for a paradigm. That is to say, something that provides fundamental explanation in terms of mechanisms of aging from first principles. But it actually doesn't do that. It's more of a kind of a list of things that happen during aging and things that are associated with aging. As my colleague João Pedro de Magalhães and I published a critique on this last year, pointing out that it is a pseudo-paradigm. And in fact, a similar sort of a model from around put forward around the same period from another group of biogerontologists called the 7 pillars of aging. It has another sort of set of things that are central to aging, and these lines that connect them sort of show how interconnected all of these different factors are. And to my mind, this expresses a sort of despair about the possibility that you could ever explain aging in terms of some fundamental causal principles. It's like everything's connected to everything else. So it's almost saying it's something that's impossible to understand because it's so complex. So another reason I think why aging has been so difficult to understand, I'd like to express in terms of a sort of a kind of analogy, which is the analogy of the TV detective series, where you very often will have some detectives trying to find a murderer. Or let's say they have a serial killer. So you have the detectives are trying to find the serial killer, and they have a number of suspects. But each of their suspects has an alibi for at least one of the killings. So they have to release each of the suspects. And the reason they can't find the serial killer is that actually there are more than one killer, right? So each one of them will have an alibi for at least one of the crimes. So what I'm getting at here is that the aging, and actually if you look at individual diseases of aging, one of the things which is very characteristic about them is that they are highly multifactorial. A lot of diseases of aging have multiple things that actually combine together to produce the disease. And in individual cases, you may have different causes that are actually more important. So each time you come up with a theory that tries to explain aging in terms of a particular thing, you can usually falsify it by showing that something else is actually important, and so your theory is wrong. Right, so what I'm going to do now is, I'm sort of jumping to my own conclusions here. I want to present, in really simple terms, the emerging model that will be the theme throughout the talk. And what I'm hoping is, in a way, by analogy, this model will act like Amundsen's choice of using dogs rather than horses to go to the South Pole to sort of lead the aging field towards an understanding of aging. So this is the sort of simplest depiction of the model that I can think of, and this is referring now. In a way, it's referring to aging in all organisms, so this applies to humans as well. So this is a very sort of high up, very generalized a way of looking at aging. But what I want to do first is to ask the question, rather than what causes aging, to ask what causes disease. What actually is disease? Why does disease happen? Obviously, there are many different causes, but one can make a generalization that a lot of diseases arise from normal function, kind of function that's specified by the normal genome, being disrupted by some factor, usually an external factor. So, for example, infection with pathogens such as viruses or bacteria or protozoan pathogens will disrupt normal function, cause disease. For example, mechanical injury like accidents, breaking bones, and so on, will disrupt normal function. Or starvation or nutritional issues like poisoning and so on will disrupt normal function. So the way that if one has a sort of ill patient, one is often looking for a cause of disruption, something that's caused the normal function to go wrong, and then you try to sort of remove it through therapy and allow healing to happen. So that's, in a way, you could think of as a sort of fundamental paradigm for thinking about where disease comes from. And the germ theory of disease is just one element of that paradigm. And one of the things that makes aging difficult is that there's actually two, really, at a very, very broad brush level, two fundamental causes that work together. One of them is the disruption itself, is disruption as just described. And a lot of aging-related diseases, disruptions can contribute to them. And disruptions have a strong effect on mean lifespan, on average lifespan. The other element, which is in a way the sort of the thing which hasn't been understood well and has been sort of underestimated, is a completely different source of disease, which is actually the normal genome, the wild-type genome, and processes that have evolved that actually become harmful in later life. And these two factors will work together to produce senescent pathologies. And a really simple one that I've just mentioned is COVID. So COVID is in many ways an aging-related disease. The SARS syndrome is very much an aging-related disease, insofar as most of the people that actually die from it are people who are elderly. And they die because they're having a massive immune response to it, which is the result of the immune system having undergone aging. So this is an example of how multiple factors work together to produce aging-related diseases. And this wild-type genotype, this wild-type genotype phenotype, this is what sets the limits to longevity of species, which is what sets the maximum lifespan. So this is why, for example, we as a species have our maximum lifespan of around about 110, whereas chimpanzees live little more than about 50 years, or mice little more than about 4 years. These are things that are encoded by our wild-type genotype. So where does the bad thing come from? Why is the wild-type genotype doing this to us? You know, it brings us into the world through development, and then it sort of takes us out again. And the answer lies in the process of evolution, particularly this principle of antagonistic pleiotropy, which I'll talk a bit more about later. So because of the nature of the evolutionary process, and particularly the presence of something called biological constraint, it leads to, inevitably, to wild-type processes that become destructive in later life. I'll come back to that a bit later. So just sort of that's the more complex version of the theory, which I'm not going to go through for time, but it's reviewed in this review that came out earlier in the year. And I just wanted to kind of credit the overall shape of this theory owes quite a lot to a largely forgotten Russian biogerontologist called Vladimir Dilman. But how the genome is actually causing aging is something that's sometimes called the hyperfunction theory, sometimes the developmental theory, but I usually call it the programmatic theory, where evolution, and particularly the reduction in the force of selection in later life, and the nature of biological constraint, that means you can't optimize everything at once, leads to genotypes that cause disease later, working through these programmatic mechanisms, which I'll talk about more later. And the ideas are not mainly from me, they're originating from particularly George Williams, the evolutionary biologist, Dilman, who was a medical biogerontologist, and more recently, Misha Blagascloni and João Pedro de Magalhães, who really developed the current version of the programmatic theory. So I think that this is really good news, because to me it's having worked in this field for a long time with a sense of not really having any sense of how you could explain things in any fundamental way. This theory looks like it has explanatory power, which is really great in terms of being able to explain origins of aging-related diseases. So now I'm going to go for the bad news, and this comes more into sort of worm territory. So this is more for the worm people in the audience. This is a little bit of history for me, really, how I got into all of this. This goes back to the early 90s. This is a study published in Nature from the lab of Cynthia Kenyon, who was at UCSF at the time. So this is a survival curve with the percentage of animals alive over time. These are worms. These are wild-type normal worms here. And here we have a mutant worms with a single DNA base change, actually, in a gene called DAF2, doubling lifespan. And I came across this when I was a graduate student. I was flabbergasted by this, because the implications are absolutely extraordinary. I mean, it's long been known that really aging is very much genetically controlled. It's controlled by, it's a trait like hair color or morphology and so on. But what it implies is that there must exist some sort of core mechanisms of aging that control the whole aging process, right? So all of these, there's scores and scores of aging-related diseases, Alzheimer's, COPD, the things that I listed, that somehow kind of above them, before them, is some central aging process, and you can manipulate that process, and you can provide protection against the whole lot of diseases. So it's like having antibiotics for aging, where you just have a single pill that will protect against all diseases of aging. That's the implication of this work. But also there's just the understanding that you could, particularly because this is in C. elegans, which is a really, really simple organism, to be able to actually understand aging and find ways of treating it ought to be actually quite easy, right? These things only live for like 2 or 3 weeks. So I just thought, wow, that is really amazing. So I moved over to Missouri to work with Don, as you heard. But this was a long time ago. I thought this would be 5 years. So this, just to put things in perspective, at that point, Gro Harlem Brundtland was Prime Minister here. Some younger people wouldn't even remember her. And for those of you who know Ariana Grande, that was the year she was born. And in fact, at the beginning, things really did move very quickly, as I would have thought, because the genes were cloned and sequenced. And the question was, do they have human homologs? And they do. It turns out this is a very simplified version of the pathway. The receptor is actually similar to the IGF-1 receptor in humans, and similar to the insulin receptor. It's the worm's equivalent of the insulin and IGF pathways. And very simplified, it works through a transcription factor, a FOXO transcription factor called DAF16. So it's actually, the pathway is switching that factor off. So if you turn the pathway down, you activate this, you get this suppression of aging. So DAF16 was sort of, in a way, became the key question was, what does DAF16 do? The secret of aging lies in understanding what DAF16 does. So what happened first was that when I moved back to UCL, thanks to collaborators at UCL, Linda Partridge working with flies and Dominic Withers with the mouse, we were able to show that this pathway is actually controlling aging in flies and in mice. So this is an evolutionarily conserved pathway controlling aging. Other groups in the States worked in parallel to us to show this too. So this was really exciting, but the question that I really wanted to understand was, so what is aging? Where's this big understanding? What's at the poles? Is there a continent at the poles of knowledge that you can tap? So it was supposed to be easy, but these genes were sequenced more than 20 years ago, and I realized about 10 years ago, I thought, if we're not getting the answers now, that's telling us something. It's telling us that we're doing something wrong. We must be doing something wrong, because the field, the talks got more and more dull. More and more genes were found, but you never got answers to these main questions. So the work that my lab's been doing over the last decade is very much just trying to do things completely differently. Look at new ideas, look at new paradigms, look at new approaches. So this is not, I haven't sort of, out of perversity, decided to do things differently and explore different paradigms. It's because I felt the field was simply failing. So experimental approaches, I'll mention this briefly. This is very much for the worm people. So this is where things got stuck. Really, in the early noughties, like 2003, we had these regulatory proteins working through some sort of effector genes, and there's masses of work being done on this, huge numbers of papers. But the question then was, what's happening between here and here? What is this aging process? So the new approach is based on a kind of a hypothesis about the steps between here and here, the chains in the cause, the links in the chain of causation, if you like. And you can see whether you think this is feasible, plausible. So what we argued is that the reason why the worms die of old age is because they have life-limiting diseases. So they die of old age because they have diseases. And so these diseases you can understand in terms of some sort of pathological changes, some senescent pathologies. And these arise because of some sort of pathogenetic mechanisms that give rise to these pathologies. And it's these mechanisms that are being affected by these regulated genes. So we have lots of knowledge here, but hardly any knowledge in this area. So my lab's been exploring this kind of unknown area, really asking questions about each of these steps. What do the worms die from? What about death itself in the worm? You know, what is organismal death? That's a fascinating area. What are the pathologies? What do they get when they get old? You know, worms get all sorts of interesting diseases actually when they get old. But the critical question really is, you know, this is the key question. What are the pathogenetic mechanisms? If you understand that, then you've got the answer to your question. So we call this the pathology-centered approach. The previous approach has tended to do molecular biology and then look at lifespan and then sort of hope that some answer would come out of that. This is arguing that you need to study the diseases to be able to make any sense of this. So in terms of aging paradigm, this is our question. What's the mechanism regulated by DAF16? So as I said, the theory, the go-to theory for many decades, which I worked on for at least a decade testing this, was this molecular damage accumulation theory. So here the maintenance mechanisms will protect against molecular damage and sort of slow down the aging process. And this is what proved to be very much inadequate. So what about the alternatives? So this is moving us towards the programmatic theory. This is based very much on ideas from George Williams. And he argued that, and others argued that aging is not an adaptation. There's no benefit evolutionarily to aging. It evolves as a side effect of natural selection in favor of where you have variants of genes that are beneficial when you're younger, but they cause harm when you're older. So, for example, my dad died from prostate cancer, and prostate cancer is something that if you don't have any testosterone, you don't get. It's promoted, it's dependent on the presence of testosterone. So plausibly, what's happened is that you've had selection for sperm production, which promotes your fitness in terms of having more children. But in later life, those benefits actually are coupled to developing prostate cancer, right? So it's not that prostate cancer has evolved because it's good in any way. It's simply a byproduct. It's because you can't optimize all biological traits simultaneously because things are all so interconnected in biological systems, as they are in life, actually. According to Williams's theory, what he says is it's action of wild-type genes, which is the main cause of aging. It's not disruption in the normal sense. It's a completely different type of cause. It's like normal disease, is a phrase that Vladimir Dillman uses to describe it. For example, DAF2, this gene that I showed you, you knock it down and you get a huge increase in lifespan. This gene is causing aging. The question is, this is the thing which has been the big question for decades, is really, we kind of know the evolutionary theory, but we don't know what the proximate mechanisms are. We know the ultimate evolutionary mechanisms, but we don't know the actual sort of biochemical and cellular mechanisms of what exactly is going on. But Williams himself, although he didn't think a lot about this, he did come up with he had an idea about it that he mentioned quite briefly in one paper, which was, he imagined a kind of like a structural gene to illustrate his thinking for calcium deposition, okay? So we have a new allele, maybe it's a gain of function, and what that does is that it sends calcium into bone, in the developing bone faster. So imagine in Africa, on the Serengeti, a gazelle, new gazelle, its bones grow faster, and so it can run more quickly and escape from cheetahs. So it's fitter. So natural selection favors that allele, but later the same allele causes calcium to go into arteries, and it causes arteriosclerosis, so it contributes to disease. So in this case, what you can see is that the gene is actively causing the disease. And this idea was developed, was taken and expanded by Blagosklony to try to explain a lot of recent findings in the field. So the pathways that were being found that control aging, where you reduce the function and you get these increases in lifespan, were very much involved in development and growth. So targeted rapamycin, IGF-1, that's insulin-like growth factor, it promotes growth. So these pathways are actually, they're pushing forward developmental and reproductive programs which will involve hundreds, thousands of different genes. And what Blagosklony argued is that these developmental reproductive programs turn into what he called quasi-programs to drive the development of pathologies. So what he means by quasi-program is that it's programmed by the genome, but it isn't a program for aging. It's not actually a purposeful program to make it happen, right? So it's because when you talk of programmed in biology, it can imply that it's a program for something, it's a function. But the evolutionary theory says that these, like prostate cancer, it's maybe kind of programmed by your genome, but it's not actually it's not a program for prostate cancer. It's just a byproduct. So Blagosklony argued that the primary cause, he was very polemical about this, that the damage maintenance theory was misguided, that actually molecular damage is mainly secondary effect of aging, mainly. It plays a minor role, but a minor role. But instead, it's actually gene action, or what he called hyperfunction. So this is in a way to contrast with the older view, which is in a way that aging reflects a kind of loss of function, a kind of wearing out. Whereas here, the genes are actively making you age, so hyperfunction. So to cut a very long story short, I'm going to spare you a lot of worm data, but we've been testing the hyperfunction theory, the programmatic theory, looking at pathologies of aging in worms. These are different worm pathologies, and essentially seeing whether we can understand them in terms of essentially good programs that have gone bad, or good processes that have become harmful processes. And this has been like the difference between day and night compared to the damage theory. The damage theory, we got negative data for about a decade. And in this case, we're starting to get wonderful explanations. And I'll just give an example. So here, I'm going to show you some worm diseases of aging. So this is the front end of a worm. Here's the mouth, and this is the structure here you can see is the intestine of the worm. And that little red line, that's the lumen of the intestine, the space inside the intestine. So here's a sort of an elderly worm. This is probably about 10 days old. And you can see here you can see the intestine. This is the lumen now. It's really big. And this is the cytoplasm of the intestine. Most of the intestines disappeared. It's just atrophied away. And these look like pools of olive oil. These are oily pools of yolk that's been produced and filled up the body cavity. So these are two different sorts of pathology. So what we found, again cutting a long story short, is we found that, so this is a sort of a section through the intestine. There's the worm, this is the intestine. So one of the things that the intestine does is that it makes yolk for the eggs of the worm. So the yolk is made in the intestine, it crosses the body cavity, and it goes into the unfertilized eggs, and then it goes into the embryos to support their growth. But what happens as the worms get older is that the worms start to consume the biomass of the intestine in order to support continued yolk production. So there's a gut to yolk biomass conversion. And what that does eventually is to cause the intestine to atrophy away. And it also, in worms that are older, when they're not producing any more offspring, they just fill up with these yolky pools. Okay? And this whole process is promoted by this insulin IGF signaling pathway, the one that if you knock it down, you get the big increase in lifespan. So we were very excited about this because what we have is a mechanism for how like a program for making yolk turns into a quasi-program that basically causes intestinal atrophy and pools of yolk. And if you knock the yolk synthesis down, you suppress the atrophy and you extend lifespan. So it's a quasi-program. It's programmatic. It's part of the wild-type function. It's not caused by damage. So it's consistent with the programmatic theory. It's supporting the new paradigm, and it's also regulated by our pathway, and it's suppressed by DAF16. So we were very happy about this. We found an actual process of senescence, of aging, downstream of the DAF16 transcription factor, which instantiates this new theory of aging. We were really delighted. But you know, things move on. I think this is part of when you get onto the right track, things start to move and change. It's like sort of the engine starts to pull the train forward. And that work led us on to some findings which I think this is kind of where the bad news comes in for people working on C. elegans, and in a way for the whole biology of aging field. There's some bad news. And this, I've got two bits of bad news. The first is coming from particularly this is a postdoc in my lab, Karina Kern. And we were wondering whether all this production of yolk in the worms, could it be an adaptation rather than just like a futile run-on of yolk production? So these are worms that are not reproducing, right? They're post-reproductive worms, and they're making yolk. So the question is, could that be an adaptation of some sort? Well, how could it be? How could it be a benefit to make yolk if you're not having any offspring? But then Karina noticed something, which is that if you take the yolk protein, this is one of the yolk proteins, and you tag it with the green fluorescent protein, you can see the yolk in the worm. It's like it shows up as green fluorescent pools. And what she noticed was that the worms were venting the yolk out through the vulva onto the plates. So they were secreting the yolk. You can see it if you look on the surface of plates of worms when they've just stopped reproducing. You see these pools of yolk. And we started to wonder whether possibly the larvae, the young worms, actually eat this yolk. And again, I'm going to cut a very long story short. What Karina showed was that, in fact, that is indeed the case. That the worms vent the yolk, and the venting is promoted by this insulin IGF signaling pathway. And the yolk consumption promotes growth in the worms, and it also even promotes slightly the reproduction in the worms that result from them. And I'll show you a little movie here. How am I going to get it to go? Oh, there we go. All right. Oh dear. So I'm not quite sure. How do I click on that? How do I get the movie to go? Oh, there you go. That's it. Right. So what you can see here, this is a pool of yolk. Oh, it's not going. No, never mind. It doesn't want to go. Oh, that's funny. Because normally I can click on. Oh, let's try again. No, it won't go. So what this shows, it just shows like a big puff of milk coming out from the vulva. So I'm going to skip. I want to cover quite a bit of ground to the conclusions of this. So we came to the conclusion that actually the worms are exhibiting something which effectively is the same sort of thing as lactation. So it's a sort of milk feeding phenomenon. And I thought this was kind of eccentric, this idea. But then I talking to other sort of biologists, I came across other examples of milk feeding among invertebrates. So this is the tsetse fly. I didn't know this, but they actually produce milk for their offspring. And this is the Pacific beetle cockroach, which does this. So this is not unheard of. There's sort of several shocks here. I mean, the lactation was quite interesting, of course, rather fascinating, a new thing that C. elegans does. But what was sort of rather shocking for us initially was that it meant that our initial interpretation, the idea that this is a futile run-on, was probably incorrect. It's not a futile run-on at all. It's a trade-off where you've got pathologies that are coupled to something that's beneficial, so they're reproductive costs. It's what we call a costly program. It defines a type of programmatic aging. Here's where it gets shocking. When you, we'd already noticed quite a lot of similarities between the pattern of aging pathologies in worms and that in a certain type of process that leads to death, which is traditionally not regarded as aging, which is reproductive death. So, for example, in a famous example is Pacific salmon. Pacific salmon, they swim upstream, and in this massive reproductive effort, they basically kill themselves from reproductive exhaustion. And in fact, if you take salmon just before they go into the spawning run and you castrate them, you remove the gonad, you can suppress these changes, and you can extend their lifespan from a few weeks up to 3, 4, 5 years. So you can, of course, if you have a programmatic mechanism that leads to rapid death, and then you block that, of course you get a big increase in lifespan, right? We've been exploring this further, and we have further evidence that strongly supports this idea that actually C. elegans are dying from reproductive death. And if you prevent the reproductive death, of course, you get a big extension in lifespan. So this was the thing that this is the shock, because maybe what it suggests is that these big increases in lifespan that we're seeing, this isn't really an extension, this isn't a deceleration of aging in the sense that we experience it. This is like preventing a Pacific salmon from spawning. So of course we see a big increase in lifespan. Something quite different. So this is a bucket of cold water, and this is the scary, that why we have this screaming Japanese lady there. So does this mean that that we should all, the C. elegans biologists, should all just sort of, go and throw themselves off a cliff in despair? Does it mean that C. elegans is a really bad model? So I'm a sort of known for being very critical of things in the field. And I can promise you that if I thought so, I would sort of grimly, with grim humor, I would say, we've wasted our time. But I actually don't think so. And here's why. What we think, and this is supported by some conceptual research that we published recently, is that actually, if you look at the different forms of reproduction in the animal kingdom, traditionally, the reproductive patterns have separated between the semelparous, like the salmon, like the marsupial mouse that I showed you, that kind of do this reproductive suicide, this reproductive death, and then iteroparous species, like our own species, like the mouse, like the fruit fly, that can reproduce multiple times. Okay, and so the idea is that in a way, this is iteroparous organisms kind of show aging. And semelparous organisms show something completely unrelated. So for this reason, plant senescence, like leaf senescence or the senescence of monocarpic plants, like annual plants, is regarded as something quite different to aging. So people who work on these things don't get invited to aging conferences, really. But what we argue is that, in fact, this view is wrong, that, in fact, there is a continuum across animal and plant species between the iteroparous pattern of aging and the semelparous pattern of aging and the way that they die. And this is supported by life history biology. The idea that there's some sort of fundamental difference between the two is not regarded as sound. And let me just give an example here. So this is I mentioned this intestinal program consuming the intestine of the worm. It's a costly program, okay? And there's an example of a costly program in mammals, in humans, or you can go right down to mice here. It's during lactation. So there's a very strong selective pressure during lactation to produce milk that will keep the young alive. And so what the mother does is to actually consume her own bones in order to release calcium into the serum that can support calcium rich milk that will allow the progeny to grow. And the bones become fragile, they fracture more easily. And then, but at the end of, so you get transient osteoporosis. But at the end of lactation, the bone grows back again. So it's a reversible cost. Okay, so that's an example of a costly program, which is quite mild in iteroparous organisms. In semoparous organisms, what you get is costly programs which are irreversible and terminal. So they change from being reversible into something terminal. So this is the gut atrophy supporting the production of yolk and progeny fitness. And that leads you to reproductive death. So the working model, and this is particularly emphasized, this is a shorter version, this is a longer version of this explanation, is that the patterns of aging that you see in things like Pacific salmon or European eel, for example, lampreys, is a kind of an exaggerated, souped-up version of some aspects of aging that exist in iteroparous organisms. So we can learn from C. elegans about those mechanisms, right? So, so in other words, then, C. elegans, it's not off in a world of its own, unrelated to aging in higher animals. All right, I'm going to mention this very briefly. This is the other shocking story. This is all published for a little while now, so I won't emphasize. I just want to give you the take-home message here. It's less important, really. This is an incredible result. Look at this. This is from the University of Arkansas, and this is wild-type C. elegans here. And these are two mutants with a mutation in a gene called AGE1, and they're living 10 times longer than wild-type. So these would be, you know, like sort of 700 years old if they were human beings, obviously. Enormous. Or no, what am I talking? 1,000 years old. So what does this tell us? What is this telling us? One possibility is that, you know, it's telling us there are core mechanisms causing the whole of aging that have been affected here. But another possibility is that, and this is certainly something that some worm people, mostly who are not familiar with evolutionary theory, kind of wondered about, which is that it's just a self-destruct mechanism. It's somehow beneficial. It's like programmed cell death, but on the organismal level. So there's a program to just destroy the worm. I initially was very reluctant to accept this idea because it's not consistent with the evolutionary theory. But to cut a very long story short, from talking particularly with Jen Law, who's an evolutionary biologist, and looking at things very carefully, we realized that actually in the case of C. elegans, unlike this would not apply to fruit flies or mice or humans, but because of certain particular features of C. elegans, the fact that they're colonial and the fact that the populations are clonal, means that actually adaptive death could evolve. It's possible, and this is actually completely consistent with normal evolutionary, the standard evolutionary theory. So, for example, adaptive death can work by, as in Pacific salmon, the salmon die and then their corpses get consumed by the baby fish. So this is what we call biomass sacrifice. Or in C. elegans, we think that it's we've modeled this in the computer, that it's because by getting rid of older individuals, they're not feeding, and that leaves more food available for individuals that can reproduce. So that actually promotes the fitness of the colony. So I'm not really going through the arguments here, but they're available if you want to look at them in these reviews. So one possibility is that insulin signaling is actually increasing fitness by promoting programmed adaptive death. So the evolutionary theory allows this for C. elegans, not for most other animal species. So this sort of this adaptive death has been explored, for example, in colonial yeast and bacteria mostly. So it's a rare, relatively rare phenomenon. And we've argued in this article that it could also have evolved in Pacific salmon. All right, so that's sort of the conclusions for the whole talk. Really, the good news is that we have this nascent theory which is emerging, which looks actually really good. I mean, I found it amazing in the last few years, a sense of things being able to put things into place in a field which has been like being lost in the fog for decades. The bad news is that the hope that we had for C. elegans, that it would provide some means of discovering some fantastic central sort of spring of aging that we could control and manipulate, the prospects for that don't look so good. And I felt a bit sad about that. And it sort of, I'm reminded by this of experience from when I was a little boy, when I was crazy about these DC and Marvel comics, you know, things like Superman and Spider-Man and the Green Lantern and especially Batman. I was very keen on. I had a Batman suit, you know, and I can remember the time when I guess I was sort of getting more self-aware, where I was sort of thinking, do they really exist somewhere, or is this just a fantasy here, you know? And I can remember being really pained by this, thinking, oh no, is this stuff really only in comics? In Batman, I reasoned that Batman probably could exist because he didn't seem to have any special superpowers. But in the end, I even gave up on Batman, that he wasn't real, and it was sad. And in a way, this thing that brought me over to Missouri all those years ago, in a way, I think it's a bit similar. It was a sort of a phantasm because of the fact that the worms are actually semelparous and showing reproductive death. But on the other hand, I do still think that we can learn a great deal from C. elegans. I'm still very excited about the work and the way that it's continuing to reveal these general principles and sort of in a way show the bigger landscape of the biology of aging. I'm writing a book at the moment about this, so hopefully in about a year and a half this book will come out. So I think what I was going to do was to try to answer the question, since we've got a question, so let's answer it. I mean, there are questions where you can answer them and you can say, yes, this is the answer, and other ones where you can make your best guess. So I guess I'm doing a bit of both here. So is aging inevitable? Well, no, it's not, because there are some organisms that show no aging. The Hydra vulgaris, they've been, I think the longest they were kept for was about 8 years. And it was estimated during that time that they would have changed every cell in their body about 120 times. And they show no signs of senescence at all. But that we don't care about Hydra. What we really want is, we want to know, is human aging inevitable, right? And that's an interesting question. Is it inevitable? Like could you actually, by that I mean, the question means, could you actually get rid of it completely? Is that possible? That's an interesting question. I think my answer to that, if you ask me, and this is very much here where this is futurology and so on and so on, for complicated reasons, I would say it isn't actually. I think it's interesting to think about what would be the conditions for actually getting rid of it. How long would it take historically? Are we talking about eliminating it in the next 100 years or whatever? I think that's a fascinating question, and sort of on reflection, I would say a very long time. But I think that the thing about aging is that it causes disease. So there will always be an ethical imperative to try to find out ways to work against aging. It will never get away. It'll never, even if we live 500 years and people after 500 years develop dementia and cancer, we're never going to be able to say, well, they've lived long enough, let them have cancer. We're never going to be able to do that. It's never going to be right to do that. So I think in a way, we're stuck with research on aging. It has to be good. But the conditions under which that would happen, it's complex. I don't want to kind of waste time going into it. It's too many things to think about. But I think a critical issue there is, of course, it depends on whether our species would survive that long. And I think there's a fundamental question about that. I think it really may not be ever possible. But if it is possible, if we could have a sort of a world government and have a sort of a control over world population in some way, have some sane policy. You know, Norway seems like, coming to Norway after Britain's in a terrible state, you know. It's like coming to a sort of sanity. So imagine a sort of like a world which is like a sort of giant Norway with a world government, you know, so that people like Xi Jinping and Putin just are an impossibility, you know, that could you know, start wars and so on. I think eventually it's likely, and I think it would be possible. So that's all. And I don't know if anyone has any questions. I hope I wasn't too long. I wasn't watching the clock.
It's worth listening to. I'm really impressed in, not the least because, as I learned this morning, that your computer was confused with somebody else's.
I found it. Actually, it was a woman who went on holiday to Portugal. She phoned up and said she got my laptop.
Yeah,
so
but
I'm feeling in a good mood.
It didn't prevent you from having to reinvent your talk last night.
Well, I didn't go to the Munch Museum, which I had been hoping to do. Just sit all afternoon.
So
reconstructing
it
the
was
talk.
very interesting, although it makes it even more difficult to understand how we can achieve healthy aging, a longer health span rather than a longer lifespan. We'll get those two to approximate. I suppose there are questions, and we have one here.
Hi, Dr. James. Thank you very much for the talk. Thomas Schmauk Medina from Oslo University in the Fanglag. So you mentioned you are excited about this new paradigm. So am I. I really like the hyperfunctional theory. But we already had one before, and several actually. For example, the Harman theory of radical species. So what in this particular theory makes you more excited than the ones we previously had? I see you mentioned predictions from IGF-1 or TOR, but even when this theory was initially proposed, we already knew these things. We already knew they were quite powerful. So yeah, and another quick one, how do you move forward to actually study this experimentally, aging itself? So regarding antagonistic pleiotropy, there might be an insane number of pathogenic mechanisms, and looking at other species like hydras or clonal trees is perhaps not realistic for us. Yeah, thank you.
Well, I mean, the history of the biology of aging has been one of kind of like a accumulating mountain of different theories. And experimental gerontology is really the field where one tests those theories. It's been like a kind of continuous process of testing theories. And I think, in a way, when I came into the field, the impression that I gained from my colleagues was that the free radical theory, the oxidative damage theory, was kind of a done deal. You know, it was treated as if it was a fact. And it took me quite a while to realize that it really wasn't. It was a kind of like a groupthink among the people within the field. It's very difficult to have a field without a uniting paradigm. Paradigms kind of unite fields. I mean, imagine if you're working in neurobiology and there are like sort of groups of different people sort of with completely different ideas about what neurons are, you know. It would be very difficult. So I think that the 90s and the noughties were a period of intense, well, they were a period of growth in the field, but also intense testing of the oxidative damage theory and the free radical theory. And my own work, I mean, I published probably about a dozen papers testing in different ways the oxidative damage theory in C. elegans with negative results. I think by about 1989, there was a kind of like a watershed in the field where I think work in different labs around the world was just not supporting the theory at all. So the reason why that theory sort of was never completely disproven, it was just that it failed to find support, let's say, was because it was tested and it wasn't useful. It didn't explain anything. In terms of the fact that the TOR and the TOR and insulin signaling pathways were discovered around in the noughties, at the beginning of the noughties at that point. But the question was, how do they actually affect aging? What does this mean about aging? And initially, for about 10 years, the emphasis was the kind of what people were doing was to try to see how these pathways control maintenance. The assumption was, aging is all about maintenance and all about damage. So these pathways must be controlling damage. What the programmatic theory has done is to suggest actually they're working in a completely different way. So what I've been doing is looking at that initially in C. elegans and showing that you can find pathologies that you can explain in terms of programmatic theory. The Ross theory, the oxidative damage theory, never explained the origins of disease. But what we've been doing lately in the lab is a lot of conceptual research looking at diseases of human aging, because what you can do is you can look at the literature and try to understand, try to see whether you can make sense of existing diseases. from the programmatic theory, and you certainly can. I mean, it's a huge task, and I've just sort of can only scratch the surface of it, but it's, people can talk to me about it later because it takes a long time to go through it all. But I think you can, it works much better. It actually, this is what the theory of aging needs to do. It needs to provide explanations for where diseases of aging come from in some general way. I think that was only your first question, actually, but I've now already forgotten what your second one was.
Yeah, so, oops, sorry. That regarding, connected to antagonistic pleiotropy, there might be an infinite number, well, not infinite,
right, yeah, okay,
yeah.
okay. Yeah, it was that. I think, yeah, I think that if you, I would say that if you look at complex metazoa, let's say everything from nematodes, insects, up to vertebrates, I mean, it looks good in terms of things being conserved in a general way, because what the programmatic theory is really arguing is that there are constraints in metazoan biology that lead persistently to the appearance of different kinds of disease. And so what you would expect is that you see manifestations of the same sort of principles of etiology right across the animal kingdom. And certainly, looking at C. elegans and the C. elegans pathologies, you can find deep homologies between them. So, for example, the uterine tumors, that is one of the most striking pathologies in the worms, and in some contexts is actually life-limiting, are actually very closely related to ovarian teratomas in mammals. They arise from the same sort of problem. So I think that when you get beyond that, it gets difficult. I think when you get to things like yeast and bacteria and so on, it's more difficult. And with plants, I'm not sure either. I think in a way, the further you go, the more difficult it becomes. But I mean, the general kind of an axiom or a kind of working theory, working part of the theory for the programmatic theory, is that there are a limited number. There's a limited sort of toolbox, if you like, of different types of etiology that give rise to these programmatic diseases across the whole of the animal metazoa. So, in other words, it's not like infinite number of different causes. That there is a sort of deep structure to it all, and especially because the programmatic theory so much is viewing diseases of aging as developmental in nature. So the developmental mechanisms are conserved, and so the diseases are conserved. If you think of something like, for example, osteoarthritis. So osteoarthritis has evolved all across the animal kingdom, right down to teleosts. You see some osteoarthritis. So this is a genetically determined thing, presumably existing because of constraints and trade-offs and antagonistic pleiotropy. The same disease recurs again and again and again, all the way down. So in a way, that gives you a sense of it's not like every bit of aging is different. So that's an optimistic view. So I'm optimistic about the theory. I think the theory is looking great. Once you get the basic theory really set, then you're into the realm of like neurobiology, where you can tear forward with huge progress instead of this sort of lost quality that the aging field has.
Thank you very much. The last question here.
Thank you, Professor, for a really great talk. So my question is, would it be fair to say that adaptive or reproductive death is not actually common for long-living species? And if it's actually the case, why we still can find any like public mechanisms in long-lived species? Yeah.
Right. So the theory, which is in the slide that I showed with the breastfeeding and the worm, the working model is that somal parity, reproductive death, is kind of derived. These organisms have evolved from species that are iteroparous. Right? It's sort of somal parity kind of appears periodically here and there around the phylogenetic tree. So the idea is that it's evolved by exaggeration. of those mechanisms. So for example, if you look at Pacific salmon that are undergoing reproductive death, and you look at the pathologies there are clearly similarities with aging in higher animals. For example, they get a pathology in the brain that resembles Alzheimer's. They get amyloid plaques. Yeah, so this is semelparous reproductive death, but the fish are getting amyloid plaques in the brain. So, and similarly, if you look, for example, at the vasculature, you see vascular changes, which are somewhat similar to atherosclerosis. You see things like muscle wasting and so on. So it's kind of a weird and exaggerated version of things that are happening in higher animals. So the idea is that the thing about the semelparous organisms, because they have this plasticity in aging, you can study them experimentally in a way which is very difficult to study aging in heteroparous organisms. So you can learn clues, fundamental clues, from them. Yeah. So for example, the whole programmatic aging model kind of was to some extent coming from C. elegans. Obviously, semelparous organisms aging is going to be very programmatic, right? But that's providing clues for aging in general.
Thanks very much indeed. We have now a little while where we can get some coffee and some refreshments. But before that, Linda has a few words to say, both of the speakers.
I'm sure that Fridtjof Nansen would be proud celebrating his birthday. And we have this tradition on Nansen Neuroscience Lecture to give this book, the biography of Nansen, that's made by Roland Huntford. And I'm sad to say that I forgot one of the book in my flat, meaning I have problems with my memory. I'm sorry, but I make sure you will get, both of you will get a book. So you come long distance, is that fair? And we will talk to each other. Yes, so, but we will see each other in December. But I will also run back to the flat and come to your hotel, so don't worry. But this is for you. And no, you get a book by your own. And by then we are closing this session, and later today there will be other talks about Nansen. And thank you, Henriette and David, for your talks.